So what is bioinformatics? Bioinformatics is the use of computers and data analysis to understand biological information, especially from genes and DNA. Bioinformatics helps us understand the role of genes in human disease by analyzing biological data to uncover genetic causes, predict disease risk, and develop targeted treatments.
Bioinformatics can answer many questions about the role of genes in human disease. Many kinds of biological information are available to help in this process of discovery, including DNA sequences, gene expression or activity measures, protein structures, folding, and interaction effects. We also have rich collections of data on the human phenotype, and indeed every health record contains such data, example, blood pressure and height. Then the fundamental task in bioinformatics is to use computational methods to connect these biological measures with phenotypic measures so that we understand how changes in one lead to changes in the other. Functional genomics, name given to the overall enterprise of deconstructing the genome and assigning biological function to genes. Mycoplasma genitalium, the smallest prokaryotic pathogen, was synthesized. Examples of some of the questions that can be asked are as follows. Structure of DNA is composed of two complementary strands made of nucleotide bases, adenine, thymine, cytosine, and guanine, that pair specifically forming double helix structure. Second is transcription of DNA into RNA to produce proteins. DNA is transcribed into a transportable single-stranded molecule called RNA. Third is structure and processing of RNA transcripts. Genes are made up of exons and introns, and through transcription and RNA splicing. Fourth is the processing of amino acid chains. After a protein is synthesized, it must be directed to its correct location in the cell, which is often determined by a signal peptide and its amino acid sequence. Fifth is epigenetic factors and altered gene expression in normal development and disease. These epigenetic changes, which can affect whether a gene is turned on or off without altering the DNA sequence, play key roles in normal cellular function and disease processes like cancer. Last is mutation is the cause of both normal variations and genetic disease. Mutation changes in DNA sequences can cause both normal genetic variation and disease, with the most common being base pair substitutions that may or may not affect protein function.
High-throughput sequencing has allowed the large-scale discovery of genes and genomes. So, how did this happen? Back in 1975, Fred Sanger invented DNA sequencing, and since then, the number of human gene sequences we've identified has grown at an incredible rate. Then, in 2000s, high-throughput sequencing was developed. This new tech made sequencing more affordable and accessible. It used to be something only a few major international research centers could do, but now it's become a routine part of pathology services. Because of that, we now have full DNA sequences for thousands of species and many human genomes. Interestingly, we are currently producing gene data faster than we can actually understand it. In other words, the amount of raw genomic information is outpacing our ability to figure out what it all means. So, here on the right side of the slideshow is a graph that represents this. The DNA data, which is represented by the light purple line, suddenly increases at around the year 2007, while the literature data, which is represented by the dark purple line, is close to being surpassed by the raw data at the year 2010. The black line represents the cost of sequencing 1 million base pairs. Back in 2000, it used to cost $10,000 per million base pairs, and now in 2010, it costs $1 per million base pairs. So all this DNA data needs to be connected to human biology to be useful, but doing that properly is only possible with advanced computer systems and smart algorithms. Even just storing and organizing this huge amount of data is a big challenge.
Gene sequence alignment methods are needed to assemble DNA fragments into plausible sequences as well as to compare sequences. Sequencing technologies. Sanger sequencing gives long, high-quality reads. High-throughput sequencing gives shorter, slightly lower-quality reads. Billions of short reads can be reproduced quickly and cost-effectively. Sequence assembly, DNA sequences are cut into fragments and then reassembled. Key steps to overlap detection, fragments merging, and creation of the shortest super sequence. Sequence alignment aligns DNA or protein sequences to find similarities. Two main types. You have global alignment, which aligns the entire series best for similar length sequences, and local alignment, which focuses on regions that have the most matches better for dissimilar sequences. Algorithms used is the global Needleman and Wunsch, local is the Smith-Waterman, and fast and heuristic is the BLAST, searches for common patterns faster but not always optimal. Application of alignment. Mapping sequence reads to a reference genome. Assembling reads into contigs, continuous sequences. Metagenomic data analysis, multiple organisms in a sample. Gene prediction or annotation using known databases, e.g. with BLAST. Gene prediction methods. Database comparison, e.g. BLAST against known genes. A/B initial gene calling based on statistical models like Markov's chain and machine learning. Data file formats. FASTA stores basic sequence into single letter codes. FASTQ includes sequence quality scores. SAM/BAM stores alignment data. SAM is readable, BAM is compressed, and VCF stores only sequence variations, e.g. S and PS, relative to a reference genome.
Decoding genomes, the role of sequence alignment. Gene sequence alignment is a fundamental computational technique crucial for understanding and working with DNA information obtained from sequencing. Neither Sanger nor high-throughput sequencing can read an entire genome at once. They produce shorter DNA fragments called reads. Sequence alignment methods are necessary to piece these reads together into plausible, longer contiguous sequences, effectively reconstructing the genome. So for comparison, alignment also allows the comparison of different DNA or protein sequences to identify similarities. These similarities can reveal functional relationships, structural insights, and evolutionary connections between genes and organisms. The alignment process, finding overlap and similarity. Sequence alignment involves identifying regions of overlap between fragments, or similarity between different sequences. So for assembly, the process involves calculating the degree of overlap between DNA fragments and merging those with the largest overlap iteratively to form longer sequences. Pairwise alignment would be comparing two sequences, and this can be global, which is aligning the entire length of similar sequences, including potential gaps, or local, identifying shorter stretches of high similarity with sequences. So different algorithms would be performed and alignments can be computationally intensive. So heuristic methods are for large databases, and they are faster, approximate methods like a blast, and are often used to quickly find regions of significant similarity. Key applications of sequence alignment. Sequence alignment is a versatile technique with numerous applications in bioinformatics. First, genome assembly, combining short sequence reads into a complete or near complete genome sequence. This is crucial for understanding the genetic makeup of organisms. So first would be mapping reads to a reference genome, aligning newly sequenced reads into an existing well annotated genome to identify variations or to quantify gene expression. Metagenomics, analyzing genetic material from environmental samples containing DNA from multiple organisms by aligning reads to different reference genomes or by assembling de novo. Gene prediction or annotation, identifying potential genes within a DNA sequence by comparing it to known gene sequences in databases using alignment tools. Methods can also use sequence features for prediction. Essential sequence data formats. Standard file formats are used to store and exchange sequence data and alignment information. So here are the different data formats.
Bioinformatics is dependent on maintaining publicly accessible research and clinical databases. So, public databases are the cornerstone of bioinformatics. They provide centralized, curated, and openly accessible repositories for biological data, including DNA and protein sequences. These databases are not isolated. Major platforms such as GenBank, EMBL, EBI, and DDBJ synchronize daily to ensure global consistency and collaboration. This open data sharing model accelerates scientific discovery and fosters transparency in research. Reference genomes are meticulously created sequences used as standard baselines for genomic comparisons. When new genomes are sequenced, bioinformaticians identify the variants, such as insertions, deletions, or point mutations, relative to these references. Many of these differences are minor, but some can have profound biological implications. Through tools like microarrays and expression analysis, researchers assess the potential impact of variants. Clinical databases such as OMIM further contextualize these variants by linking them to known genetic disorders. To summarize everything that was discussed in the last two slides, we have key takeaways. So, in summary, public databases are indispensable in the field of bioinformatics. They provide the infrastructure for storing, accessing, and analyzing vast biological data sets. Reference genomes simplify the process of identifying meaningful variations, while bioinformatic tools and clinical databases bridge the gap between raw data and real world medical applications.
Detecting how specific genetic patterns affect disease was difficult, especially when many genes are involved, so they turned to functional genomics and microarrays to make things easier. Functional genomic studies directly measure the activity of the genes in a particular biochemical mechanism. Microarrays, or DNA chips, contain a high-density array of hundreds of thousands of DNA probes. These probes bind to predefined DNA patterns, allowing us to test for gene expression across an entire genome in one experiment. Here's how it works. We extract mRNA from a sample, like a tumor or healthy tissue, and convert it into complementary DNA or cDNA, labeled with fluorescent dyes. In this diagram, we use dark purple for reference and light purple for the tumor. This labeled cDNA, known as a sample probe, is then hybridized to a slide containing a grid of single-stranded cDNAs called probes. After hybridization, the microarray is scanned with a laser. The brightness at each spot corresponds to the amount of RNA that was present in the original sample. Light purple spots indicate genes that are upregulated in the tumor. Dark purple means downregulated, and gray shows equal expression in both samples. Why microarrays matter? This technology lets us simultaneously measure the activity of many genes. It's useful comparing gene expression under different conditions, like healthy versus diseased tissue, or between different organisms. Microarrays have made it possible to uncover patterns in gene activity that were previously undetectable, giving us powerful insights into diseases at a molecular level.
Today, I will explain about machine learning methods assist in the discovery of association between genetic and phenotypic data. Genomic studies generate data from tens of thousands of genes but often have only few samples. This makes the data high-dimensional and difficult to analyze using traditional methods. Machine learning solves this by detecting patterns in large, complex datasets. These are two main types. We have supervised learning and unsupervised learning. Supervised learning, which use data to predict outcomes like disease types, and unsupervised learning, which finds hidden patterns or clusters in the data. Gene clusters helps reveal how genes are related. Tightly grouped genes often work together in the same biological process, while scattered ones may be related. Machine learning can also integrate other types of information like gene expression, clinical records, and environmental data over time. This helps scientists understand how genetic features influence health, traits, and disease. Overall, ML is a powerful tool in making sense of complex genetic data and supporting discoveries in precision medicine.
While we sometimes identify single genes responsible for diseases, the reality is far more complex. Genes rarely work alone. Scientists now study what we call the interactome, the vast network of genetic and molecular interactions shaping biological processes. When studying, we often find numerous genes associated with a single condition. The challenge lies in understanding each gene's specific contribution. Genes may correlate with a disease for several reasons. First, they might be structurally co-located. Driver mutations that directly cause diseases often appear alongside harmless passenger mutations. Second, genes might regulate each other. One gene's protein might activate another gene. Third, multiple genes might participate in the same biochemical pathway, all necessary for a specific function. How do we unravel these networks? Bioinformatics help infer relationships from expression data. We conduct genetic experiments like knockout studies and model organisms to see how removing one gene affects others. Time series studies are particularly valuable, showing how gene expression changes as pathways activate. This temporal ordering provides crucial clues about causal relationships. Machine learning now plays a vital role in modeling these networks. Boolean networks, Bayesian approaches, and Markov models help represent gene interactions. With so many biological entities potentially involved, selecting the right features become critical for accurately modeling. Understanding these complex networks ultimately brings us closer to understand and treating disease. While Human Genome Project has revolutionized biology by enabling computational approaches to many biological questions, we must guard against what could be called computational triumphalism, the belief that all disease-related problems will now be rapidly solved through computation alone. This optimism rests on three questionable assumptions. First, genetic reductionism, the belief that all biological regulation can be inferred from DNA sequences alone. Many computational approaches assume we can predict physiological changes from nucleotide alterations. However, cellular metabolic pathways involve multiple complex interactions that extend far beyond simple genetic coding. Second, the computability assumption that the belief that we can accurately model complex biochemical phenomena like protein folding. Computer science research suggests that protein folding is NP-hard, likely computational and solvable. Yet we still aim to predict interactions among thousands of molecules, from RNA transcription to protein function. Third, the closed world assumption, the belief that our mechanistic model are complete and reliable, while projects like eCell attempts to model cellular functions based on known regulatory behaviors, our knowledge remains incomplete. We now understand that environment factors after epigenetic controls significantly influencing disease risk, what we call the diseasome. The gene network visualization from ALS shown in the figure exemplifies both the power and the challenge. We can map connections between genes, but fully understanding the biological context of these relationships remains elusive. Bioinformatics, though originally a subspecialty of health informatics, has evolved largely independently. However, with the growing reliance on computational tools like machine learning and the integration of phenotypic data from electronic health records, or EHR, the two fields are now closely interconnected. Clinically oriented bioinformatics relies on health terminologies, ontologies, and EHRs, while health informatics must increasingly manage genomic and other omic data to support genetically informed clinical decision-making. Consequently, clinical systems, including decision support tools, protocols, vocabularies, and patient records, must integrate advancements from bioinformatics and genomic medicine.